we're back with Colin. Wow, that feels so interesting to say. I feel like I'm like a news anchor or something. But well, so thank you again for doing this. I'm really excited to kind of dig through. I was saying that it's quite surprising because, or not surprising, but like we've actually covered a lot of the questions I had. So then for the rest of this, I wanted to go through like the few questions that I have left, and then have Colin do whatever he needs to do to explain the rest of the UI for onboarding purposes. So yeah, if you want to take it away.
Sure. So we're just going to take a look at a different data set here, just to give us a different idea of what the interface can look like. And this one has a different feature as well, which it has these X's. And so this each one of these, if we hover over them, let's see, you can see that it says vaccine selected 2014 925, and this one up here says vaccine selected 2017. So if we scroll up to here, vaccine selected 2019 927, and so part of what the lab does is to study the flu. And if we scroll down here, this is a different visualization that you haven't seen yet. This is I think the word for this is a cladogram, which is a kind of a neat let's see, I believe cladogram is the word, and I might be wrong about that. Let's see. Oh nope, I am. Okay, I think it's maybe a Muller, might be a Muller plot. Okay, let me try one more thing.
Okay.
Oh, Muller plot is no longer Googleable. Oh no. Okay, hold on. Okay, let's see. Muller plot. There we go. Let's see. Is this it? It is. Okay, here we go. So for everyone following us from the visualization side, this is a Muller plot. And this is a tree. This is basically which over time which clade has more population than the other. And so if we look at this, we can see that this maps this, like, let's look at this more recent one, A1B 137F. That's the newer one, and that has maybe 30%. And we can look up here 137F, right? So that's like we can highlight those.
Sorry, can you define clade?
Yes, clade is part of tree. So yeah, let's go to the Wikipedia page for that too. So clade is a group of organisms that consists of a common ancestor and all its lineal descendants. Related to like taxonomy. There you go. This is all right. Again, related to phylogenetic tree. You have a phylogenetic tree. A part of that tree is a clade, and so this is if we go back to coronavirus here, we don't necessarily that you can't color by clade on. Oh, wait a second. I take it back. You can color by clade. I haven't done that yet.
Oh.
Okay. So, and then if we open that up, yeah. So the clade is a part of that's meaningful to the scientists, but not necessarily to lay people. But the idea of a clade, the idea that the tree kind of has parts, it's kind of interesting here because it partly explains, like, if you get a flu vaccine and it didn't work. Maybe you got a strain which was kind of far away. There are multiple strains of the flu circulating, so and then you can kind of see the relative prevalence of which countries have one strain of flu relative to the other, and you want to. It gives some view into the complexity of making public health decisions around like who you might want to vaccinate for what.
Yeah.
Kind of a neat. This is a neat and different view into the tool. Something that it can do that we didn't really cover before. As I thought we might come back with this, just to give everyone a really different view. You can see where the vaccines were and what they were picked, and like the vaccines corresponding to a strain, because you have to take a strain and you have to kind of inoculate the eggs. So growing flu vaccine in eggs is something that is kind of a medical industrial process. That let's see. Oh, there you go. So. If you didn't know, so the flu vaccines are, to my understanding, grown in eggs over time. So
Wow.
Gotta pick a clade you want to defend people against. So all right. This is that's just something else. A really different way the interface looks in a different context. Like it's not so these can be pie charts. Basically, I could have said the map also can display as a pie chart to show relative frequency of clade if that's what you're showing.
And then right now for COVID-19, it's just a little bit too new to be able to color by that. So
it actually does have coloring by clade. I'm just not qualified to say what it means.
Got it. Okay.
Yeah. Meaningful to them, but not to us. We're mostly going to know, like, for lay people, it's like coloring by date or coloring by country or coloring by administrative division is meaningful. And so that's where you can see, oh, this red group here, happens to be like Washington. And then if we kind of zoom in here, we can see, U.S. Washington. These are all the sequences from Washington and U.S. California. I believe actually this gray one right here is the it might be the cruise line. Not quite.
Oh.
I think that actually is the yep, out in the water.
Yep.
So I believe that's the cruise line. Okay, so we wanted to go back to you were asking just before the break.
Nucleotide.
Yeah, right. Okay, so all right, so this is where it gets a little over where I think I'm in the most risk of saying something inaccurate. But basically, if we consider down here where it says diversity, let's go back to Ebola real quick because we can see the genomes for those. So the genome for Ebola has some 18,000 positions, and if we scroll down here, we've got we can see the letter at each position.
Yes.
Right, and those might change. And so if we go back to Nextstrain for Ebola,
the little bars are how many times it changed? Is that it?
Yeah. So exactly. So if we click on that, we can see the part of the tree where it's flipped. So this part of the tree has a different different genotype. In this case, this is not just ATC, but a group of them. So this is also an added level of complexity. We might, the tree is not, that graph is not just considering C. It's also considering, like a group of them together.
I see.
And so if that mutates, if there's a mutation that affects that, it can affect what the virus does and how it behaves and things like that. So this is also surveilling, and also not all of the parts of the genome are kind of created equally or of equal relevance to the researchers. This was also something that was surprising. Different parts of the genome are more and less relevant in terms of what a mutation what a mutation in that one place versus another might do. So that's really where it gets into the domain specific knowledge that's specific to the virus and to the scientists.
So is what you have? Sorry, saying the diversity part, but then the kind of those little bars that say MP and BP35, is that also the domain specific part of like
this part right here?
Yeah,
that's the domain specific part. Yeah, that's like this section of the genome for Ebola is responsible for doing X, Y, or Z. Or I think the thing which was kind of mind blowing to me was that part of the genome is responsible for the the shell or the outside of the virus which is where it attaches to the cell, and a mutation there can make it more or less infectious and things like that. And that's kind of where that was something which was surprising.
Got it.
So you might watch different sites with varying levels of interest, is my understanding.
I see. And I noticed that those kind of like spanned the parts where the bars exist. So that's really interesting also.
Right.
Okay, so I think I now understand that the diversity part, and I think there was a question that was, I think the other day when I was looking at the map, it was a pie chart, but some parts of the pie were gray and some parts were colored, and I had and that was one of my questions.
Yeah, right. So it's likely that what you saw was a lack of data, and so the lack of data can be because there's no information. For instance, for location, that's a good example. If we color by location, then here we do have location Boston, right? Whereas or location Wuhan. Versus just having or location Snohomish County. This would be United States as the United States as country, Washington as the administrative division, and Snohomish County as a location. And this is something that in the Nextstrain code base is kind of complex because, or sorry, it's a complexity that has to be handled because you have to find a mapping to state, province, city, and the different ways in which countries handle their administrative divisions, which are country specific. But the tree has to basically have a color by, which is general. And so country is country maps to country, administrative division maps to state or province or anything like that, and then location is the kind of the most detailed bit.
Got it. Yep. Okay. It's super helpful. Now that we're on this tree, I also noticed the different tree layouts. And so I wanted to ask you if you use any of the tree, because I think we've been on the tree layout and I think the clock before, or sorry, the rectangular and the clock. And I was wondering, and then for the clock, so then my question was what the other two, so I've noticed that when you do clock, you almost seem to exclusively also use like branch length divergence.
Right, and they're actually the same. They don't actually change it in this case.
Okay.
But time, but time does add the rate estimate of 30 subs per year. So.
So and then this is compared to Ebola, which was like 0.00105 subs per year or something like that.
Right, and that's out of my domain knowledge as to whether that implies that one is evolved. If we can actually say that means that it's evolving faster than the other, it may just mean that maybe the more samples you get that changes, or maybe overfitting to what's happened early. So
got it. So and then the question was what the radial and unrooted layouts might be good for or used for.
Sure. So let's go here to we'll stay on administrative division and we'll go to radial. So the radial tree I think would best be compared to the rectangular tree. So here we're gonna and again we'll simplify this by using time, sampling date. And so if we go from here, we can see that time goes left to right. In radial, time goes inside to outside, and the dates, the rings of the dates go inside to outside. Radial can help kind of de-emphasize. You'll notice how much area is taken up in rectangular by these early, like look at the branches and how much space these early branches take up and the branch lengths. In radial, you're going to squish all that down and you're going to kind of compress it. So it's kind of like a geographic projection that puts all the distortion, in the ocean or something like that. I don't know. That's a maybe that's an improper metaphor, but it's going to shorten the branches is the effect.
Got it. I kind of, when I was going through this, it clicked easier for me to look at the radial, I think because for me personally, in the rectangular one, when for like the further down you went, like there were so many, like so many dots that they overlapped, and this one spread it out more. So that makes sense as like what you were saying about like kind of layout and distortion.
Yes, I think also we can see that if we're going to administrative division and we look at we can see what's at the center. Here we go. Here's a good comparison. Let's look at the Washington cases, right? So we have Washington here, and the gray ones are the cruise ship administrative division Grand Princess. So we have a a group here, which is, the tree is going to kind of squish everything in one way, right? But if we go to radial, it's really going to, if we follow those over here, it kind of breaks it up. It kind of makes it clear that this is its own little group. It's kind of easier sometimes to see the clade here because of the way the branches are laid out. So.
Got it. And then the unrooted layout.
Yeah, so the unrooted layout is to me a little bit hard to understand and explain.
Okay.
My understanding of this is that by allowing, what's colored by sampling date, just to see. Yeah, right. That it allows things which might be artificially separated to kind of be squished more together, so that if you kind of see this right here, like all the Washington cases that are really similar, like if they're genetically identical, you can squish them together.
I see.
Yeah, and so I think my understanding is that this allows you to show similarity, and not because like let's take Washington, right? And if we go to administrative division, which is going to be red, and we'll see red over here. On tree, red is going to maybe, I don't know, maybe it's going to be spread out more than it would be in unrooted. It allows you to kind of squish it together more. So that I'm the least clear on how that one's.
Okay, I was going to ask a question. But based on what you said, let me ask it anyways and see if it's... so my understanding just now was like, for example, with some of the... for some of the dots, they overlap because, like you said, that they were very similar or the same, I think.
Right.
Whereas with the Washington ones, it seems like there's quite a few that are like, there seems to be some like distinct branches, and so they might be more similar. Ooh,
that's kind of a neat one, maybe to show that. I think that's like, if we go to unrooted and divergence, that's actually going to, so that's going to show, I believe that's going to show to that point. If we look at Washington all the way over on the right, that's these guys here, and we go to divergence, it's going to squish them all to a point.
I see. Okay. Okay, that's really helpful to know.
If we go back to rectangular. Oh, sorry, go ahead.
Sorry, what?
No, go ahead.
Oh, I was going to then ask about the rest of the options on the left-hand side, because I think we've covered a good chunk of like the top 3/4. And then there's like branch labels.
Yes. Right. So again, to go back to clade, we can label the tree by clade, and we can change the map resolution to country, which is going to be let's see if we color by so we can color by administrative division, and this would allow us to answer questions like, what percentage of the U.S. cases are Washington? This is not the best interface to get that kind of information. It's not like, it's not as dashboardy and specific as that. Like, you can't hover over it and see that information, but you can get a snapshot look. Also, we can color by division. And we can also color by location, or show by location, and then that can again kind of zoom in. And in that case, it'll put the U.S. cities, right, rather than if we go to division, it'll put it at the state level in the U.S., and then otherwise it can put it at the city as well. And then we do have a grid option. If you want to look at these things side by side, you can zoom in here, like on an area, and then maybe subset the tree just to Washington, something like that. And then take a screenshot. Something that facilitates that kind of sharing.
Got it. I have to admit that all of the links down, like all of those links, I thought were like citations or something, and I didn't even like look at them to realize that they were filters.
Right. So the submitting lab is here, and there's a lot of this is credits, and these do. But you can filter by originating lab. So if you just want to see like the Dutch COVID-19 response team, and you click that, then that will show you a certain number of genomes, like over here, which I believe would be probably in the Netherlands. There we go.
Wow.
So there you go. So these now we've kind of mapped just to just to those genomes sampled by the Dutch COVID-19 response team.
There was a network error.
Uh-oh. Do we lose our stream?
Okay. This is quite the adventure of a stream. Okay. Yes, we got it back now.
Okay. Great.
Okay. I think that was all of my questions. If there are any more that you want to cover for us.
I think I should show you the animation. Hold on just a second.
Yes, okay.
That's a fun one. All right, so let me go to... All right, I'll go to full, and then I'm going to remove the filter, and I'm going to go to time and color by administrative division's fine. That'll give us, we can try a couple different colorations. And then there's a couple options for coloring the map and kind of what it's going to show. And so let's see. All right, I guess this is probably best if we do this. Now we do double it. You can see that it's doubled. So that if you're looking at the U.S., if you want it to be U.S. centric, you can still see China and Europe. And if you want it to be Europe centric, you can still see U.S. and China. And if you want it to be China centric, you can still see. So we duplicate. If you zoom out, you can see the tree is duplicated and then stops, and then duplicated and then stops.
Oh wow! Okay.
So that's that. It's basically two full and I think it's two full and then a half. So. Yep. All right. So let's zoom in here. So we'll center on it on China because that's going to make the most sense for the animation. And then we'll let's see. That's good. And then we can come down here and go to, we'll say medium, and we'll just leave these default for now. Or actually, we'll go fast because it's going to be on screen share. So, okay, loop animation. So loop animation will make it go again and again. We won't do that for now, and we will animate cumulative history. So what this means is that it's going to play everything forward. And if you can see up here as a legend, it's basically doing the same thing we were doing with the time slider. Yeah. Right. It's moving the time slider forward, and as it moves the time slider forward, you can see the steps as transmissions spread to other places in the world. And then more towards the end of the tree here, there's going to be a lot of sequencing in Europe and US, obviously between the end of February and now, and then you kind of reflect that here. Yeah. Wow. Yes. Yeah. So a big jump. Really right. Big jump in sequencing at the end of February, as Western countries start testing, and then if we can see it's quite different if we reset this and then turn off animating cumulative history, and here we go. And so this is kind of yeah. So this is a time slice. So you can see if we go up here, it's animating just this bit of the date range. Okay. And as it animates that slice, you can see, it looks more like you're animating. Sometimes you have the tree, or sorry, sometimes you have the nodes and the demes. Other times you just have like a branch on the tree, and then you're animating across, like over that branch. And again, this one is sequences. This is not total cases. This is just sequences. So, I think the Dutch having 82 of the 477 really overrepresents the Netherlands. They've just done a lot of sequencing. And so it doesn't mean there's more cases in the Netherlands. If we zoom in here, but you can see it does appear that the Netherlands has done more sequencing than Italy has, for instance. And that's not that there's more cases in the Netherlands, just more sequencing. Yes. Important distinction for the entire interface. Yes, definitely. I'm just currently mesmerized by the animation. you might play it locally as well, and there's a there may be delay through the zoom. I don't know what the respective frame rates are, but yeah, we can. The steps to replicate this are pretty simple.
Yeah.
You just go to Nextstrain, and then go to the coronavirus page, and then scroll down to the bottom, and then basically just click play on the map, and the map will animate, and it's kind of equivalent to scrubbing through that time window.
Hydrosquall asks, what's an example use case for looking at two trees simultaneously? It came up in a PR review. Great
question. I will have to defer that to the biologists. I don't know. I don't understand that particular graphic.
Okay. Yeah. Okay.
Good question though.